This video tutorial is about gel electrophoresis. Electrophoresis is used to separate different sized fragments of DNA. It can separate fragments that differ by only one base pair and is widely used in gene technology to separate DNA fragments for identification and analysis. The technique uses an agarose gel plate covered by a buffer solution. Electrodes are placed in each end of the tank so that when it is connected to a power supply, an electric current can pass through the gel. DNA has an overall negative charge due to its many phosphate groups, and the fragments migrate towards the anode, which is the positive electrode. Fragments of DNA all have similar surface charge regardless of their size. The principle for separating proteins is the same as for separating DNA fragments, but is often carried out in the presence of a charged detergent, such as SDS, which equalizes the surface charge on the molecules and allows the proteins to separate as they move through the gel, according to their molecular mass. In some cases, the proteins can be separated according to mass, and then, without SDS, according to their surface charge. This technique can be used to analyze the types of hemoglobin proteins for diagnosis of certain conditions, such as sickle cell anemia, where the patient has hemoglobin S and not the hemoglobin A. A DNA probe is a short 50 to 80 nucleotide single-stranded length of DNA that is complementary to a section of DNA being investigated. The probe may be labelled using a radioactive marker, usually with 32 phosphate in one of the phosphate groups in the probe strand. Once the probe has annealed, i.e. bound, by complementary base pairing to the piece of DNA, it can be revealed by exposure to photographic film. Alternatively, the probe may be labelled using a fluorescent marker that emits a colour on exposure to UV light. Fluorescent markers may also be used in automated DNA sequencing. Probes are useful in locating specific DNA sequences, for example, to locate a specific gene needed for use in genetic engineering, or to identify the same gene in a variety of different genomes from different species when conducting genome comparison studies, or alternatively, to identify the presence or absence of a specific allele for a specific genetic disease that gives susceptibility to a particular condition. Scientists can place a number of different probes on a fixed surface, known as a DNA microarray. Applying the DNA under investigation to the surface can reveal the presence of mutated alleles that match the fixed probes, because the sample DNA will anneal to any complementary fixed probes. The same DNA must first be broken into smaller fragments, and it may also be amplified using the polymerase chain reaction, PCR. A DNA microarray can be made with fixed probes specifically for certain sequences found in mutated alleles that cause genetic diseases in the well. Reference and test DNA samples are labelled with fluorescent markers. When a test subject and a reference marker both bind to a particular probe, the scan reveals fluorescence of both colours, indicating the presence of the particular sequence in the test DNA.